Welcome to our webinar, which will be discussing the topic of infrared vision. Our presentation aims to show the benefits and influence that infrared technology would have on human vision. Our webinar is influenced by the 1987 film Predator. The film presents aliens which are able to detect light in the IR spectrum and convert it into thermal images. The vision comes to an advantage for the aliens as they are able to use thermal imaging to clearly track and target their prey during the night. The film highlights the advanced technology possessed by the aliens, as through their helmet they are able to visualize their target based off of heat and radiation. The film acts as an inspiration, as if humans were to acquire this technology, it would produce a wide range of benefits.
The human eye has the ability to detect light in the 400 to 700 nanometer wavelength range in the electromagnetic spectrum known as visible light. Visible light enters the eye through the cornea and reaches the retina, essentially stimulating the rhodopsin and photopsin photoreceptors in the rod and cone cells respectively. This visual stimulation of photoreceptors results in the formation of electrical impulses, also known as action potentials, that surge onto and stimulate the optic nerve. Upon stimulation of the optic nerve, action potentials travel through the axon and synapse with cell bodies in the visual cortex in the occipital lobe, thereby allowing us to perceive visual images from the visible light stimulation. Given humans can only detect visible light being 400 to 700 nanometers, Palchuska and colleagues actually proved otherwise, as they observed that human rhodopsin and photopsin photoreceptors can in fact detect infrared light being 800 to 1000 nanometers, which were radiated from laser beams into the eye, essentially electrically stimulating the photoreceptors and subsequently the optic nerve in the same manner as visible light, as explained in the prior slide. So the question arises, why can't we see infrared light if our retina is able to detect it? Well, all the infrared radiation emitted from the sun is either absorbed by atmospheric gas or water molecules or by the human body, since we are composed of 70% water. Moreover, the eyeball is made up of fluids known as aqueous humor that is able to absorb infrared light before it reaches the rod and cone cells of the retina in the back of the eye. The same is true for UV light, but not for visible light, as seen by the water absorption in the graph, and this explains why we can see visible light and not UV and infrared light. With this in mind, since infrared light cannot reach the eye, scientists have been researching other methods that are inspired by snakes to provide infrared vision to humans, which will be the basis of the next slide.
Several snake species are capable of observing thermal images through infrared vision. Pythons, vipers, and boas all possess pit organs on their face, which allow them to detect thermal radiation of surrounding organisms. Nerve cells in the pit organ are responsible for the detection of infrared radiation. TRPA1 ion channels contained in the nerve cells act as infrared receptors, which enable snakes to sense infrared radiation as heat. At certain temperatures, the TRPA1 channels are activated and opened. This allows ions to flow into nerve cells and trigger an electrical signal to produce thermal images of their surrounding environment. For example, at 28 degrees, the TRPA1 receptors of a rattlesnake are activated. At this temperature, the snake is able to sense the heat of a mouse from 1 meter away. A thermal image of its prey is then able to be produced.
Understood how infrared vision is achieved by snakes, we will now discuss how scientists have exploited TRP channels to optimize infrared vision in species that don't possess it. Recently, Dasha Neladova and her colleagues engineered an optogenic technology to aid those with incomplete blindness, and our technology is inspired by the ability of snakes to generate thermal images from heat radiated from infrared light. Like humans, certain snakes contain visual organs allowing them to see in visible light spectrum, but some species of snakes also contain temperature-sensitive TRP cation channels that are expressed in a specialized pit organ, allowing them to convert infrared light into thermal images, essentially allowing snakes to superimpose visual images and thermal images. Therefore, snakes are able to switch back and forth between two imaging systems individually or utilize both simultaneously. Now, in order to achieve infrared light sensitivity in organisms that don't express TRP channels, Darshanaladova and colleagues engineered a light detector on retinal cells, comprised of a genetic component being the TRP channel with an extracellular epitope, and the nanomaterial component being a gold nanorod conjugated to antibodies that bind to the epitope on TRP channels. Thus, this dual system has been designed so that the gold nanorods are able to absorb light, converting it into local heat, thereby opening TRP channels. And once the TRP channels have opened, then the infrared light current is able to access photoreceptors of cone cells. This stimulation of photoreceptors in cone cells leads to the same pathway as visual images as explained in prior slides through the optic nerve, to essentially allow the perception of thermal images. This system takes advantage of the fact that near-infrared water absorption is low, thereby allowing the activation of the implemented TRP channels in retinal cells. However, local overheating in the eye is a problem that still needs to be addressed by these scientists. Darshanaladova and colleagues implemented the dual system into blind mice and observed that blind retinal mice cells to be infrared light sensitive. This allowed the blind mice to perform behavioral tasks like drinking water. The wild-type group of mice which don't have the dual system implemented were not able to respond to infrared light seen in the graph. In addition, Darshanaladova and colleagues were able to evoke infrared light sensitivity in human ex vivo, proving that this type of technology is a novel strategy to provide therapeutic technology to humans with incomplete blindness. And in order to achieve this, the next step would involve testing in human clinical trials to prove the safety and efficacy in humans before it can be implemented to the broader population.
Engineering infrared vision could possibly restore light sensitivity in humans suffering from blindness. It would assist the visually impaired as they would be able to use heat and radiation to detect humans and other obstacles against their surrounding environment. The technology can be further implemented into humans to improve vision, as it would allow both visible images and thermal images to be observed simultaneously or switching between the two. Infrared vision would enable firefighters to see their target through smoke-filled rooms. This will improve the quality of their work, as instead of carrying around an additional thermal imaging camera, which is currently used, infrared vision would simplify this process as carrying unnecessary equipment would be reduced. It would further implement more safety on roads as detection of humans or animals at night would be made more clear, limiting the amount of danger on our roads. Medical appointments would also improve as medical professionals would be able to detect diseases, infections, and cancers due to the temperature changes occurring in the body. For example, rather than undergoing blood tests, MRIs, or X-rays to diagnose arthritis, a simple glance with infrared vision would diagnose arthritis, as the temperature changes in the body would be made more clear.